Protein synthesis consists of transcription, mRNA processing, and translation. Transcription contains 3 steps: initiation, elongation, and termination. mRNA processing consists of 5' poly-A tail, RNA splicing, and alternative splicing. The first step of protein synthesis is transcription, and the first step of transcription is initiation.
This is the RNA polymerase, this is the DNA strand, and this is the promoter region, and these are the nucleotides.
The first step in transcription is initiation.
Initiation!
After RNA polymerase binds to the promoter region, the DNA strand unwinds and the polymerase initiates RNA synthesis at the start point of the template strand.
And step 2 transcription is elongation. In elongation, the polymerase moves downstream, unwinding the DNA and elongating the RNA transcript through 5 through 3. In the wake of transcription, the DNA strands reform a double helix.
Third and last step of transcription is termination. In termination, the RNA polymerase reaches the end stop of the DNA strand, and that is when termination occurs.
The second step in protein synthesis is mRNA processing. It consists of 4 parts. First, the 5' second, poly-A tail, third, RNA splicing, and the last, alternative splicing. Before mRNA can be processed, a modified version of guanine, aka the 5' comes to the 5' end.
RNA polymerase releases the pre-mRNA. The poly-A polymerase then binds to the strand, which cuts off 3' ends and attaches a sequence of poly-A tail, which is 250 or more adenine nucleotides, helping the mature mRNA leave the nucleus and protecting the mRNA from degradation by hydrolytic enzymes, and help ribosomes attach to the 5' end of the mRNA once the mRNA reaches the cytoplasm.
During RNA splicing, introns are cut out and extrons are spliced together by spliceosomes. The third and last step of protein synthesis is translation. Translation consists of 3 steps.
1. Initiation, 2. Elongation, and 3. Termination. Initiation. A small ribosomal subunit binds to a molecule of mRNA. The mRNA binding site of this subunit recognizes a specific nucleotide sequence on the mRNA just upstream of the start codon. An initiator tRNA with the anticodon UAC base pairs with the start codon AUG. This tRNA carries the amino acid MET. The arrival of a large ribosomal subunit completes the initiation complex. GTP provides the energy for the assembly. The initiator tRNA is in the P site. The A site is available to the tRNA bearing the next amino acid.
Elongation. The anticodon of an incoming aminoacyl tRNA base pairs with the complementary mRNA codon in the A site. Hydrolysis of GTP increases the accuracy and efficiency of this step.
This step removes the polypeptide from the tRNA in the P site and attaches it to the amino acid on the tRNA in the A site.
The ribosome translocates the tRNA in the A site to the P site. At the same time, the empty tRNA in the P site is removed to the E site, where it is released. The mRNA moves along with its bound tRNAs, bringing the next codon to be translated into the A site.
When a ribosome reaches a stop codon on mRNA, the A site of the ribosome accepts a release factor.
The release factor promotes hydrolysis of the bond between the tRNA and the P site and the last amino acid of the polypeptide, thus freeing the polypeptide from the ribosome.
The two ribosomal subunits and the other components of the assembly dissociate.
The end. Goodbye.